This is the fourth screencast in the nervous system physiology series. As I said previously, we split this up into 5 sections. We've covered the first 2 sections at the moment, so I just thought I would stop and give you another or an update on the lecture aims. So the lecture aims of the next couple of screencasts is to understand neuronal synapses, to understand neurotransmitters and receptors, and to understand what we call synaptic integration and function. And this should take us probably another 2 screencasts. So screencast 4 and screencast 5 correspond to nervous system lecture 2. Okay, this is a placeholder slide that I've shown you before. We talked previously about how the nerve action potential was generated, how it propagates down the axon, the importance to some extent of these nodes of Ranvier. We'll talk about these probably later on in the course, and then how the action potential propagation into these terminal branches, these nerve terminals, is important. And what we're going to concentrate on in this screencast, the next 2 screencasts probably, is the release of neurotransmitter at these areas here. So officially this is part 3 of the 3 lecture series, neuronal synapses and receptors. So what I'd like you to do here is just have a look at this diagram, because it tells us a little bit about the different types of connections or synapses, shown here, here, here and here, that we can find within this neuronal system. We've already said that this is the neuron, this is the cell body, and these are the dendrites out here, and this blue structure is an axon. So you'll see that there are 4 main types of synapse shown here. Synapses can form between predominantly between axons and the cell body, the soma, so they're called axosomatic. We can also get axodendritic synapses from an axon onto a dendrite rather than the cell body. Let's say in slightly rarer cases, we also get axonal synapses formed between 2 of these blue axons, and we can also get dendrodendritic synapses, where a dendrite synapses makes this junction with another dendrite. So yeah, this is maybe just a point of interest before we get into talking about neuronal synapses in a little bit more detail. Probably if we'd have been having a similar lecture maybe 150, 200 years ago, and I was to ask people listening what they thought was the nature of a synapse, was the signal passed by electricity or by a chemical? Probably about 150 years ago, most people would have probably reasonably suggested that it was electricity that passed from one of these synapses to the other. So from here to here, across these synapses, there was some kind of electrical connection. In fact, what we now know is that the signal is not passed by electricity, because that would be it'd be inefficient for the body, but a chemical is used. So in the mammalian nervous system, 99% of the synapses that I showed you on the previous slide are chemical in nature. So what that means is a chemical is released across from one of these pre-synaptic elements to one of these post-synaptic elements, and it isn't electricity. As I said, 99% of our synapses use chemicals, neurotransmitters. We'll talk about some of them as we go through the next few slides. The electrical synapses are sometimes called gap junctions, and as it says here, they're just open channels that conduct electricity directly. Usually they're joined to each other or very closely opposed, rather than the gap that we see in these chemical or these specialized junctions called synapses. So 99% of the mammalian synapses are chemical in nature, and hopefully you'll understand that after the next few slides. As I said, this maybe just summarizes that previous diagram. Axons of pre-synaptic neurons make contact with target post-synaptic cells. So the post-synaptic cells can be another neuron, can be a muscle like at the motor end plate, or maybe a gland that secretes. Again, you'll find out about glands and hormone secretions in other parts of the lectures. So these are the predominant types of synapses that we have, mainly axodendritic, from an axon to a dendrite, axosomatic, from an axon to a cell body or soma, or axonal synapses. And again, we'll see the prevalence of these as we go through the next few slides. Presynaptic cells release a chemical neurotransmitter, and they act on specialist proteins on the postsynaptic, typically on the postsynaptic cell, called receptors. And again, we'll find out a little bit about some common neurotransmitters and receptors in this lecture, and this will form a basis of for going on to do much more about transmitters and receptors as you go through your course. And again, this is probably the first time you'll have seen this diagram, but you'll probably see it in my lectures again as you go through into part 2 and part 3. So it's a diagram of chemical synapse. So there's a few important things basic things to pick out here. So this is our presynaptic element. This is our postsynaptic element. So presynaptic terminal, postsynaptic cell. Neurotransmitters are typically packaged into these round bodies shown here called vesicles. Vesicles migrate to the presynaptic membrane where they fuse, and the transmitter substance that's contained within the vesicle is released across this narrow synaptic cleft. Synaptic cleft is around about 20 nanometers wide, so 20 times 10 to the minus 10 meters, very narrow, but not directly opposed like you would see with a gap junction. So the transmitter is released, goes across the synaptic cleft, and acts on these postsynaptic receptor proteins. This protein is driven by the action potential, which we talked about before, and requires energy to package the transmitter into the vesicles. That's why these mitochondria are here. So maybe just talking you through this, the action potential invades this presynaptic terminal, causes a change in voltage, because that's what the action potential does. Voltage gated, calcium channels, calcium comes down its concentration gradient, and there are calcium dependent interactions between proteins on the synaptic vesicle and the presynaptic membrane that cause the vesicle to fuse with the membrane, open and release its contents across the synaptic cleft. So voltage gated calcium channels are important. Again, some of these, bit more detail you'll find out about later on in the course, but just make sure you understand the basics for now. The vesicles fuse with the presynaptic membrane, as I said, release neurotransmitter into the cleft, which is 20 nanometers wide. The neurotransmitter acts on these postsynaptic, typically postsynaptic receptors. Sometimes there are some presynaptic receptors. Again, these are things that we'll talk about later on in the course. But make sure you understand this as the basis of what we call synaptic transmission. So what we'll do for the remainder of this screencast is talk a little bit about the first major type of neurotransmitter, those which increase nerve activity, and they're called excitatory neurotransmitters. In the second screencast, we'll talk about inhibitory neurotransmitters that decrease nerve activity. But for now, let's focus on excitatory neurotransmitters. As the name suggests, they increase nerve activity. From a, let's say, biophysical point of view, they cause depolarization, and they initiate what we call an excitatory postsynaptic potential, or an EPSP. Maybe the first important thing to point out is that EPSPs are different to action potentials. So APs are action potentials, EPSPs are excitatory postsynaptic potentials. They occur at the postsynaptic cell. And again, we'll see how this occurs as we go through the next few slides. Common examples of excitatory transmitters are things like glutamate. So glutamate is our major excitatory amino acid, and also things like acetylcholine. Acetylcholine acting at the neuromuscular junction is an important example of an excitatory neurotransmitter, We'll talk about this in these lectures and also when we talk a little bit about muscle physiology in the next set of screencasts that you have with me. So yeah, just maybe to tell you a little bit about the other type that we're going to talk about in the next screencasts. So inhibitory neurotransmitters, to contrast those with excitatory, they don't increase but they decrease nerve activity. From a biophysical point of view, they do this by not causing depolarization but causing hyperpolarization, and they initiate an inhibitory postsynaptic potential, so an IPSP. Examples of these are our most common inhibitory amino acid, so gamma aminobutyric acid, GABA. Opioids are endogenous painkillers, things like enkephalins, act as inhibitory neurotransmitters because they reduce pain signals, and acetylcholine is also inhibitory neurotransmitter. And the reason I've put this in red here is probably just to remind me to tell you that this is not a mistake. So we saw on the previous slide that acetylcholine can be an excitatory neurotransmitter. It can also be an inhibitory neurotransmitter. And this maybe is an important point. If we were doing this face to face, I'd probably ask you how that could be. So you could pause the screencast and have a think about it. How can acetylcholine be both excitatory and inhibitory? So if you have paused the screencast, you can unpause it now and I'll tell you. It's not a mistake. Basically, this is a good illustration that the neurotransmitter, the way that it acts, is dependent on the receptor that it interacts with. So as you're going to see maybe in the rest of these screencasts and certainly other parts, maybe when we go on to the next set of screencasts, there are a number of different receptors for acetylcholine. Some of them cause an excitatory response, some of them cause an inhibitory response. So there are neurotransmitter receptor subtypes. Some of them can be excitatory, some of them can be inhibitory. And again, hopefully not to confuse you, I'll just tell you that all of the subtypes that glutamate act on are excitatory. All of the subtypes that GABA act on are inhibitory, as are opioids, but acetylcholine, some of them are excitatory and some of them are inhibitory. So important take-home points: receptors dictate whether a neurotransmitter will excite the system, will increase nerve activity, or inhibit the system, decrease nerve activity. So that's probably actually a good place to take a break from this end of the first screencast. What we'll do in the next maybe two screencasts, the first one we'll definitely talk about excitatory neurotransmitters, and we might split it into two shorter screencasts. We'll see how the time goes in the next one, but that's a good place to finish the first screencast.